Hello my dear friends and a warm welcome. In this video you will get important multiple choice questions on the topic electrophoresis. Myself Dr. Tripti Malik Ahuja, I am your guide and mentor for the subject microbiology, life sciences and biology. This video will help you in preparation of your exams, university exams, BSc UG level and MSc PG level. It will also help in the preparation of competitive exams like CSIR-NET exam, GATE exam, ICMR exam and the entrance exam for the CUET PG. In the description box below, in the playlist and in the I button here, you will find there is a lecture video on the topic electrophoresis and rocket immunoelectrophoresis. You can see the video and these videos will help you in your studies. Before I start the multiple choice questions, I request you to take out a paper and pen. I will read the question and then the options. The options which you think is correct, you can note it down and then I will give you the possible explanation of the correct answer. So now let's get started. The question number 1. Which technique separates charged particles using electric field? Options are A. Hydrolysis, B. Electrophoresis, C. Protein synthesis, D. Protein denaturing. This was a bouncer question and I know you have answered it correctly. Yes, the correct answer is B. Electrophoresis. Electrophoresis means the movement of the particles under electric field. And these particles are of course the charged particles. Question number 2. Electrophoresis was developed by another easy question. The options are A. Sweat, B. Swedberg, C. Tiselius and D. Sanger. The correct one is C. Tiselius. Question number 3. Isoelectric focusing is a technique routinely used to resolve proteins based on their. This question is a previous year question. It has been asked in the DU exam, Biochemistry entrance exam 2019. Option A, hydrophobicity. Option B, mass. Option C, pi. And option D is length. The correct one is the option C, which is the pi. Pi stands for the isoelectric point. It is that pH at which the net charge on a particle is 0. So the correct answer is C, pi, isoelectric point. The question number 4. In 2D electrophoresis, purification of proteins is based on. This is another previous year question asked in the CUCET exam, Biotech 2021. Option 1, A. Molecular weight. Option B. Charge. Option C. Number of subunits. Option D. Both A and B. Here the correct one is the option D, which is both A and B. Now let's see how this is correct. In the 2D electrophoresis, there is two methods, or we can say there are the two steps of separation. The first step is based on the isoelectric point. That means it is isoelectric focusing, and the second step is the based on the size. So that step is the step of the SDS page. So this is the two-step focusing. This is based on the SDS page second step. So first step on the basis of the isoelectric point, that is a pH. That second is on the basis of the size. The question number 5th, for the separation of DNA by electrophoresis, which of the following method is commonly used? This is very simple question. I hope you can answer it correctly. The A is Agarose vertical. B option Agarose horizontal. C option page vertical. D option is the page horizontal. The correct one is the B, Agarose horizontal. Agarose gel electrophoresis, which is in the horizontal manner. Question number 6. If proteins are separated according to their electrophoretic mobility, then the type of electrophoresis is? Another straightforward question. The option A, SDS PAGE. Option B, affinity electrophoresis. C, electrofocusing. D, free flow electrophoresis. The first one is correct. SDS PAGE, sodium dodecyl sulfate PAGE. Question number 7. The electrophoretic mobility, denoted as mu, is mathematically expressed as. This has been asked in the previous years in number of exams. The option A, VE, B, E oblique V, C, 1 oblique EV, D is the V oblique E. The correct one is the option D. The mobility, it is expressed as the rate of migration divided by the electric field. The next question, question number 8. Which of the following factors does not influence electrophoretic mobility? Asked in CUET Microbiology Exam 2019. Option A, molecular weight. Option B, shape of molecule. C, size of molecule. And D is stereochemistry of molecule. The option D, stereochemistry of the molecule, is the correct. It does not depend upon the conformation. Question number 9. Which of the following statement is true about migration of biomolecules? Option A. You have to tell the statement which is true. The option A, rate of migration is directly proportional to the resistance of medium. Option B, rate of migration is directly proportional to current. Option C, low voltage is used for separation of high mass molecule. The option D, rate of migration is inversely proportional to the current. Here the correct one is the option B, rate of migration is directly proportional to current. Question number 10. Which of the following molecules, biomolecules, are not separated by electrophoresis? That means out of this biomolecule, one molecule is that which we cannot separate using the electrophoresis. I hope you are writing your correct option. The option A, separation of protein. Separation of amino acid. C, separation of lipid. D, separation of nucleic acid. The option C is correct. The electrophoresis is not used for the lipids. Question number 11. Agarose can be extracted from which of the following? A, Lycasusium esculentum, Ficum benglensis. C, Gracilaria esculentis. D, Agrotis stolonifera. The option which is correct is the option C. It is Gracilaria esculentis. It is a red algae, and agarose is a polymer of galactopyranose. Question number 12. Which of the following cannot be used for separation of nucleic acids? A, SDS PAGE, B, only page, C, northern blotting, and D, horizontal electrophoresis. Option A, SDS PAGE, is the correct. It is used for separation of proteins. Question number 13. Which of the following statements about agarose gel electrophoresis is false? Now note very carefully that out of the 4 statements which are given here, there is one statement which is incorrect, that means which is false. So the option A, electrophoresis in agarose is a common way to separate DNA molecules according to size. This is correct. Ethidium bromide is used to detect DNA. It is also correct. Under the influence of electrical field, positively charged DNA will migrate towards the negatively charged end of gel. Now see, it is written positively charged DNA, but DNA is negatively charged, so it is incorrect. The fourth, none of them. So here the correct one is the option C. Question number 14. The polymerization of the gel used in page occurs between polyacrylamide and A, N-acrylamide, B, bisacrylamide, C, N-methylene acrylamide, and D, N-methylene bisacrylamide. The option is correct is the D, N-methylene bisacrylamide. Question 15. The fluorescent dye such as Ethidium is used for visualizing DNA. How do Ethidium binds to DNA? A, stacked between histone molecules. B, binds to the nucleotide base. C, intercalated between the stacked bases. And D, binds to the phosphodiester backbone. So the C is the correct, intercalated between the stacked bases. Question number 16 says, if 2% weight by volume of agarose in gel is used, what is the effective separation size range of double-stranded DNA? Option A, 0.1 to 3 kb, B, 0.5 to 7 kb, 1 to 20 kb, D, 5 to 60 kb. The option which is correct is the option A. It is the 0.1 to the 3 kb. So this size is used for if the size is the 0.1 to 3 kb, it can be separated easily by using the 2% weight by volume. This is chart showing if we want 2 to 30 we can use 0.5, then we want 0.75. If we want 0.7 to 20 we used 0.75. So we can see there is a gradation of the percentage of the agarose according to the size. If we are requiring the lesser sizes, so we are using the more concentration of agarose. Another related question is question number 17. Nusieve agarose is designed for analytical electrophoresis for resolution of DNA fragment. Now see, this is a very recent question, not included in most of the textbooks. This Nusieve agarose is a recently added type of the agarose, and the option is that A, small DNA, RNA and PCR products lesser than the 1 kb, B, used for obtaining protein bands, C, column chromatography, D, ion exchange chromatography. This Nusieve agarose, it is used for the very smaller DNA, RNA and the PCR products which are very small, lesser than the 1 kb of the size. Question number 17. The sodium dodecyl sulfate SDS used in SDS page is very simple, straightforward question, but it has been asked in the CUET exam, Odisha exam 2018. An anionic detergent, B, a cationic detergent, C, a non-ionic detergent, and D is it is an ion exchanger. So I hope you have guessed it correct. Option A is correct. It is an anionic detergent. Next question number 18. In SDS page, migration of protein is affected by A, charge of the protein, B, size of protein, C, net charge of protein, D, all of this. The option B, size of protein, is correct. Question number 19. The role of APS in SDS page is 2. APS stands for ammonium persulfate. The options are A, acts as a catalyst in the polymerization of acrylamide, B, act as a source of free radicals, C, act as a bridge between acrylamide and bisacrylamide, D, acts as a pore builder in the polymerized gel. The option B, it acts as a source of free radicals. It is an oxidizing agent and it is often used with another agent which is timid. Timid stands for tetramethylene ethylene diamine. It catalyzes the polymerization of acrylamide and bisacrylamide so that the polyacrylamide gels they are prepared for the electrophoresis. The question number 20. The pH of 1 stacking gel, 2 resolving gel, 3 tank buffer in SDS page is respectively. The option 1. 8.3, 8.8, 6.8. Next option 6.8, 8.8, 8.3. Third option 8.3, 6.8, 8.8. Last option any of the above. Here the correct answer is the option B. The stacking gel, it is slightly acidic. Then the resolving gel, it is basic, while the tank buffer is also basic. You have to remember all of this pH. Question number 21. The tracking dye used in the SDS page will be the option A, anionic, option B, cationic, option C, non-anionic, option D is amphipathic. The correct one is the option A. It is anionic. It is anionic, hence it will migrate with the leading edge of the proteins being separated on the gel, and it is the protein being a negatively charged molecule, hence it moves towards the positive electrode. Question 22. Electrophoresis of histones and myoglobin under non-denaturing conditions. pH is equal to 7 results in both proteins migrate to anode. The option A. Both proteins migrate to anode. Option B. The histones migrate to anode and myoglobin migrate to cathode. C. Histone migrate to cathode and myoglobin migrates to anode. The option D. Both protein migrate to cathode. Now for this question we have to see for the isoelectric pH of these two proteins. The histone, it has the pi of the histone is 8.5, while that of the myoglobin is the 5.5. Under denaturing conditions means when the pH is 7, it is a neutral pH, the histones they will have the positive charge while the myoglobin will have the negative charge. So the histone, it will migrate to the cathode and the myoglobin will migrate to the anode. So the option C is correct here. Question number 23. In SDS page, the protein sample is first A. Treated with reducing agent and then with anionic detergent followed by the fractionation by electrophoresis. B. Fractionated by electrophoresis then treated with an oxidizing agent followed by the anionic detergent. C. Treated with an oxidizing agent and then with the anionic detergent followed by fractionation by electrophoresis. D. None of the above. So here the option which is correct is A. It is treated first of all with the reducing agent like the mercaptothenol and then with the anionic detergent like the SDS page, then it is fractionated according to the size. Question number 24. Which of the following is used as a tracking dye in SDS page of protein? Option A, bromophenol blue, B, xylene xylenol, C, orange, G, anti, all of these, they can be used as the tracking dye. So the D option is correct. Question number 25, the last question. Ethidium bromide is a stain that specifically binds to DNA or RNA and appears dash. Dash means the color is asked if viewed under the ultraviolet, right, UV light, red, yellow, orange and blue. So here the correct option is the option C, which is the orange. So these were the 25 questions which you will find helpful for your preparation of the exams. That's all for now. Bye and take care. Best wishes.